My name is Huda Zahir. My name is Huda Zahir, PhD student in Hassan First University, Faculty of Science and Technology, Department of Biology, and I prepare my thesis on antimicrobial activity of the medicinal plants in Morocco. So today I can start my presentation. Hello, good morning.
Hello, we are hearing you. Yes.
I hope you can hear me very well.
Yes, we can.
Very well.
So I can share my presentation.
Yes, sure, please. Share your screen.
So can I start?
Yes, please. We are here listening to you.
So can I start?
Yes, sure. You can start.
Okay. So good morning, ladies and gentlemen. Today I will be giving a short presentation on evaluation of the antibacterial activity of extracts of the local medicinal plant, Opuntia ficus indica, on the growth of multi-resistant strains. So firstly, I want to thank my professors Tawsi and Bouchaib Ben Sharqi for their helps in this project, and also the members of scientific committee for giving me this chance to participate with you. The plan of this presentation, I will start by a short introduction, moving on materials and methods, results and discussion, and I will conclude. Firstly, what's the meaning of antibiotherapy? Simply, it's a treatment by an antibiotic or an antibacterian. It's an organic chemical substance of natural or synthetic origin that inhibits or kills pathogenic bacteria at low concentrations and has selective toxicity. Selective toxicity means that it should be toxic just for the bacteria and not for the cells infected. The concept of antibiotic resistance is the ability of a microorganism to resist the effects of antibiotics. There is several types of resistance. And this study, we were focused on multi-drug resistance bacteria, which carrying several resistance genes for different antibiotics. So the fight against antibiotic resistance is now a medical and also an economic emergency. Not in this quickly, bacteria resistance will cripple our health system. The WHO estimates that in 25, the 10 million people will be died because of this resistance. If no new antibiotics are developed, there is a risk of one day being faced with bacteria against which no known antibiotic will be effective. The situation requires finding new medical economic based and natural product presented by a new mechanism of action against bacteria. So the plant tested in this study is the prickly pear, or such as her scientific name, Opuntia ficus indica, belongs on genus Opuntia and family Cactaceae. So this species is widely cultivated in arid and semi-arid areas of Morocco and other parts of the world, and reported to have sound biological activity. We were trying to test different factors that can be influence the compound composition, like the different parts as cladode and flower. And also we tested different variety were collected in 2 different regions of Morocco. And also we tested 2 stages of vegetative development, pre-flowering and flowering. The prickly pear is a natural remedy that is effective in the fight against different diseases such as cholesterol, diabetes, and cancer. The sampling was carrying out with a cactiton of Jamiat Riyah installed in the International Institute of Research Agronomic. This cactiton contains 33 ecotypes or variety of these species. We were collected in different regions of Morocco, such as Jad, Nata, Oued Hidou, etc. We were focused on 2 variety of Bjad and variety of Gulmi. The cladodes and flowers were harvested during 2 stages between January and May, pre-flowering and flowering, and these samples were put in plastic bags and immediately placed in a cooler. Each sample was cut into thin slices and dried in an oven at a temperature of 50 degrees Celsius for 72 hours. Grinding of the plant is an important step. Can you hear me, sir?
Yes.
Hello?
Yes.
Yes, no
You can
problem.
hear me?
Yeah, sure.
Grading of the plant is an important step that the plant will present a larger surface contact with extraction solvents, thus improving the yield of extractions. So the method of extractions used in this study is a hot maceration with the SOX leaf. The principle of this extraction or the advantage that the condensed solvent accumulates in a siphon tank, which increases the contact time between the solvent and the powder. When the solvent reaches a certain level, it primes the siphon and returns to the flask within the dissolved substance. So it was a successive maceration with the solvent of increasing polarity. We use hexane for eliminate the fat and lipophilic substance. And then we use the acetone to extract the medium polarity molecules, and finally ethanol to extract the polar molecules. Each extract was or concentrated by a rotavapor according to the boiling temperature of each solvent. The strains studied in this work come from the ATCC referenced collection. We prefer to use different strains bacterial because each one has a specific metabolism and structure. We used 2 strains gram-negative, Escherichia coli and Pseudomonas aeruginosa, and 3 strains gram-positive, Staphylococcus aureus, Bacillus cereus, and Micrococcus leachus. So the analysis of antibacterial activity led us to definition of different activity parameters. First one is qualitative evaluation. The principle is the preselection sensitive bacteria. It's called also sensitivity test by the disk diffusion method. The second one is quantitative evaluation by determination of MIC or minimum inhibition concentration and minimum bactericide concentration using the liquid dilution method, and the medium used in the study is Miller-Hansen, either solid or liquid. So for preparation of the unicolumn, colonies well separated from the bacterial culture already subcultured are taken, homogenized with 2.5 ml of sterile distilled water with vortex, and we obtained bacterial suspension. For inoculation, we spread 15 microliter for each strains with tight streaks over the entire agar surface. Moving on preparation disk. So firstly, we prepare a concentrated extract at 600 milligram per ml. We take 6 milligram for each extract and homogenize it with extract solvent. The nitrocellulose paper disc, 6 millimeter in diameter, are impregnated with the different volumes, 10 to 20 and 30 microliter, and deposit of the surface of plates, kept 1 hour for pre-diffusion, and we place it in for incubation during 24 hours at 37 degrees Celsius. The interpretation was made by measuring the diameter of the inhibition zone around each disk. To determine the MIC in a series of 6 tubes numbered from C1 to C6, we add 1.5 ml of MHEB medium, and we add 1 ml of each extract to C1 with vortex, and we transfer an ml from C1 to tube C2. We continue like this until tube C6. And we add in sterile way 0.1 ml of bacterial suspensions to each tube. And finally, we obtain a dilution series spreads out as follows. We add also a control negative and control positive. And after incubation from 18 to 24 hours, the MIC is the lowest concentration of the substance for which there is no growth visible. To determine determine MBC, we use a bactericid control 24 hours early, and we inoculated in each plate like this series of dilution from 100% to 0.01%. After the type MIC or the type which there is no bacterial growth, we inoculated with the Strix and incubated. And finally, we compare the Strix to the bactericid control. And the MBC is the smallest concentration whose subculturing shows a germ growth less than or equal to 0.01% of survival. Ratio MBC per MIC is a characteristic for each antibacterian or antibiotic. So we can say that bacteriostatic when the MBC per MIC ratio is greater than 4. And we can see that it is bactericidal, means that can kill bacteria when the ratio is less than 4. We also test the effect of temperature. We use the extract that show a very good activity. In this case, it was the ethanolic extract. We treat it with different temperatures, 13, 37 and 55 and 100 and 121 degrees Celsius for 1 hour. And the same method we use it for determine the diameter of zone, the inhibition zone. Moving on results and discussion, diameters of zone of inhibition of the different concentration tested with the two controls for the 5 meter resistance. So as you can see in these graphs, the diameter depends the concentration used, and also depends the strains tested in this study. And we also notice that the variety of jet shows a very good activity compared to variety of Gilliam. And also the flowering state and part flower showed a very good activity compared to other fractions. So as you see, is in most cases the gram positive was the most sensitive to the extract compared to gram negative bacteria. and thus we can explain by membrane permeability barrier that limits access of the antibacterial agents to their targets. For the ethanolic extract, showed interesting and very good activity compared to acetonic extract, as you can see here. Always the variety Ubijade showed a good activity, and we can also notice that the control positive we use it didn't affect the bacteria Pseudomonas aeruginosa compared to our extracts that show a very good activity. This bacteria is very resistant. It's really very resistant bacteria. So for ratio MBC per MIC, the both of our extracts, the acetonic and the ethanolic, showed a bactericidal power for the 5 germs. Concerning the effect of temperature, it didn't show any effect on ethanolic fraction. On the contrary, it showed a very interesting activity, and that reveals that there is a thermostable molecules, and also that justify our choice to hot maceration like a method of extraction. For conclusion, that our results shows that ethanolic extract has a good effect compared to acetonic extract, and we can focus on it to determine or to develop and characteristic compound composition for future studies. Thank you for your attention, and I hope you enjoy it.
Thank you very much, dear Hoda Zaher, for your presentation. Dear participants, is there anyone who has any question to dear Zahir or any contribution? Okay, thank you very much, dear Hoda Zaher. By the way, your certificates, probably until tomorrow, you will receive your certificates, presentation certificates. For all authors, we are submitting certificates. Now, for next presentation, from the research group, Forestry Research Institute of Nigeria, any member of this research group with us? Because I can't see the whole screen. Let me read the names. Oluwayesimi, Emmanuel Toyinbo, Titulop Adeleke, Babatunda Olajide, Francis Ojo. Anyone from this research group with us? I think there is no one.
I don't think she's here yet. Maybe we can proceed while she joins us.
Okay, okay. And the next presenter, dear Abu Fadeju, Jonathan.
The same team. She's also a member of the same team.
Yes, yes. Okay, thank you very much. And from University of Uyo, Florence Abolaji Bello, Rosa Itatom. No.
Yes, I'm with you.
Hello,
Yes.
Are you here?
Yes, I'm here.
Welcome, how are you?
Fine, thank you.
That's great. So, okay, we can start. Please introduce yourself shortly and you can start your presentation.
Okay, I want to share my slide.
Sure, sure. And let me, dear Zahir,
Please kindly
end your presentation, stop your presentation, screen sharing, because we see your desktop now.
Okay.
Yes, you
can still not share.
Dear Hoda Zahir, is that desktop belongs to you?
Which one?
Excuse me, dear Bello, is that desktop now on screen? belongs to you?
No.
No, okay, because it belongs to dear Zahir. Dear Zahir, are you hearing me?
Yeah, sure. Yes, I can hear you.
Yes, please stop your screen share because we see your desktop now.
Okay, I will try.
Yes, yes, dear Professor Bello, you can share your screen now.
Yeah. Okay. All right. Please, I'm still trying to connect.
Yes, no problem. We are waiting for you.
Okay. Okay. Please, have you found it there?
Mhm.
Have you seen it?
Now we can't see your share. So I think there is a problem. Dear Florence Bello, can't you share your screen?
Sorry, sir. May I suggest maybe we take the next person while
Yes,
she's
Hello,
sharing
sir.
her screen?
yes, we'd better if you do it. Dear Samson, Oluwatosin Taiwo, Hamza Abdul. Now next to you, you can start your presentation. Then we'll
Okay.
be back.
Thank you very much.
Thank you.
Okay.
Mhm.
Good day, everyone. Hello.
Hello, hello.
Can you hear me?
Hello.
We can hear you. We can hear you.
Perfect. Okay. So I want to begin the presentation. Good day, everyone. This morning I will be presenting a topic entitled cassava flour addition to yam flour, improved functional, color, and textural properties of Pando yam flour. Now, I'll be doing this presentation on behalf of my team members, Tosin and Amza. This is my presentation outline. I'll be taking you through this. And to your right-hand side, you can see a nice meal. Unfortunately, we cannot eat this. It's a sumptuous meal made from yam and vegetable soup. But maybe going forward, when there is an advancement in 3D printing, we may begin to print this out and everybody can have a bite. All right. Now, what is pounded yam? Pounded yam is a traditional food in Africa, but it's most widely consumed in West and Central Africa, and it's majorly made from yam. Now, it's a glutinous food that is made by pounding yam in mortar and pestle, resulting into a very smooth, consistent dough, like you can see on the plate to your right-hand side. Now, the process for doing this is very tedious, and it's also very time-consuming. I'm going to be showing you a video shortly to see how this is actually prepared. And I'd like you to follow. It's a very short one. so that those who are not familiar with this would perhaps understand how this is produced. We can follow, just you'll be looking at this quickly. Now, the video you're seeing right there, that is how pounded yam is made traditionally. And it is very tedious. Okay, fine. I'm going to stop that video, then I'm going to continue with my presentation. So that is how pounded yam is made. It's really, really a very tedious process. And so because of this strenuous method of making pounded yam, researchers came together to find an alternative. And the alternative is to produce the yam by pre-cooking it into chips and then drying in the oven and then making it into flour. Now we have different types of pounded yam flour in the market, different brands, as you can see on the right-hand side. Now, but because of the high production, because of the technology involved and the high production of the flour, efforts are being made to find a way to reduce the cost. And this is the thrust of our research. Basically, previous research attempted to fabricate facilities for processing the yam into flour. Other studies focus on improving the nutritional value of the pounded yam. It will be important to note at this point that pounded yam flour is strictly majorly carbohydrate. In 2005, Oluwamukomi and Adeyemi reported 5 times increase in the protein content of pounded yam flour that was enriched with defatted soybean flour. Now, in 2010, Babajide also attempted to improve the textural properties of pounded yam. I say quickly that one of the important attributes of pounded yam, the dough, is the viscoelastic properties that the consumers actually cherish when they consume this delicacy. Now, Babajide also further found that sensory properties of the dough improved when cassava flour was supplemented at 20% level. However, there are currently no studies on the functional and the textural properties of cassava flour yam flour supplemented with cassava. So what then is the aim of this study? Now, the basic aim, we have a broader aim, but for the purpose of this conference, we are looking at the proximate composition, color, and functional properties of pounded yam flour composited with cassava flour. Now, you've seen the image right there. We can have yam, and to your right-hand side is the cassava. Now, these were obtained from farms and markets in Nigeria. Figure 1 quickly shows the process flow chart for producing pounded yam and cassava flour. The materials were sourced, peeled, and diced into chips ranging between 0.2 to 0.3 centimeter. Now, they were soaked in methyl sulfide solution for 30 minutes. And the essence of this is to prevent browning reactions because once you cut these tubers and they're exposed to oxygen, browning sets in. So, but when you soak them in such solutions, it helps to delay the onset of browning. And then the chips were dried, milled, and then packaged. Okay, now the methods or the analysis that were conducted on the flour include proximate composition, the functional properties, and the dough was prepared according to Babajide et al. 2010, the color of the flour and cooked dough, and then textural properties. Now, samples were prepared in duplicates and analysis were done in triplicates. Now, again, I'd like to show you when the flour has been formed, I'd like to show you how the dough is prepared now from the flour sample. Quickly, I'll be showing that also. I want you to follow. For those who do not know how this is done, basically, we'll look at that quickly in another 2 minutes. Now, you place water on fire. As soon as the water is boiled, you get your flour samples. The flour samples is poured. You can see that. You pour it into the pot, and then you make a dough by stirring. You keep stirring. Now, this is different from the traditional method where you have to pound and use your energy. Now, that's what you do. You keep mixing it until you get a consistent dough. You allow it to simmer, and then you stop. Now, because of time again, I would want to stop the share and continue with my presentation. So that is what we do basically to get your dough without going through the stress of pounding, which is typical of the traditional method. Now, So now table 1 shows the proximate composition of the pounder yam flour. Now, what you have noted, I'd like you to draw your attention to the fiber content. Generally, with increase in level of cassava flour, the ash, fiber, and carbohydrate content increase. And obviously, this can be attributed to we can attribute this basically to the high content of carbohydrates in the cassava flour. If you look at the table, cassava has 85.3% carbohydrate compared to the yam flour, which is 80.83%. So the addition of cassava flour obviously enhances the carbohydrate content and also the fiber content of the flour. And of course, this is important because pounder yam is a starchy product. So the starch content of the cassava enhanced the property of the proximate composition of table 2 shows the color of pounder yam flour supplemented with cassava flour and its cooked dough. Now, to my left-hand side, you would see that generally the L values, which indicate lightness, were higher than those of the dough. And these can be attributed to browning reactions and as a result of the heat that was applied during the mixing of the dough on fire. Now, the total color change for the flour was not significant when cassava was added at 15% level, and the same trend was observed for the dough. Table, if you also look at the next slide, figure 1 or figure 2 shows the functional properties of the pounder yam flour. 4 properties were evaluated. The first one is the bulk density, the water absorption capacity, digestibility, dispersibility in water, and the swelling power. Now, bulk density tells you how compact your sample is. And generally, if you observe, you would see that the yam flour was more compact than the cassava flour. And also, the loose bulk density generally were lower than their packed bulk density. And obviously, this is as a result of the removal of the air during tapping of the flour samples in the tube. And also, we observed that the water absorption capacity of the flours increased with increase in levels of the cassava flour. And previous studies showed that water absorption is a function of the granule size, the amylose content, and of course, the high fiber content of the flour, as I showed in the previous slide, may have accounted for the increase in the water absorption capacity of the flour. The swelling power also increased with the increase in temperature. And of course, the increase may be due to the gelatinization of starch in the flour component. And this has been attributed to the changes within the flour component, including the starch, that may have enhanced the increase in swelling power. Table 3 shows the texture profile of the dough. Basically, when the dough was formed, a thickness of about 20 to 30 mm was made on a texture meter, and then the hardness, springiness, adhesiveness, cohesiveness, and gumminess properties of the dough were evaluated. Now, if you look at table 3, generally, there was an increase in the hardness of the dough with increasing levels of cassava flour. And of course, this is expected because remember, like I said, the carbohydrate content of cassava starch was higher than that of the yam flour. And obviously, previous studies showed that cassava has higher starch content than yam flour. And this may explain the higher hardness value for the dough with increasing level of cassava flour. It's also important to know that the adhesiveness also increased from 0.95 to about 5.92 Newton seconds. Now, in the increase in adhesiveness, of course, which means that the thicker suggests that the supplemented flour had improved textural properties. Babaji et al. In 2010 suggested that a stickier dough is an important property of pounded yam. And obviously, cassava has enhanced the textural properties of this dough. Now, to conclude my presentation, the composite flours were more dispersible in water compared to the yam and cassava. And this may be attributed, like I said, to the starch content of the cassava flour. The addition of cassava flour also increased water absorption capacity and the lightness of the pounder yam flour and the corresponding cooked dough. Thirdly, samples with cassava flour were firmer, more sticky, and gummier than the control dough. And this is, of course, an overall improvement in the textural properties of the dough. Now, we recommend that a pilot scale model may be required to scale up these current findings, and a thorough economic analysis is also required. Why? Because remember, at the beginning, one of the justification of this study is the cost, which currently is associated with the purchase of pounder yam flour in the market. And obviously, if this is done, we can reduce the cost drastically and then enhance the production of the flour. This will provide convenience and also allow more people, including those in the rural area, to buy the pounder yam flour with their money. These are selected references from this presentation. I want to thank you for your attention, and my team would like to appreciate the Comrade State University and other sponsors for giving us the opportunity to participate in this conference.
Dear professors, thank you very much for your presentation, and thank you very much for your participation with your students. Now, dear Florence Bello, are you ready? A couple of minutes ago, I think you have a small technical problem. Now, could you solve it? Dear Florence Bello, are you hearing me? Dear Florence Bello, are you hearing me? No voice. Dear Professor Samson, if you want, you can proceed to your other presentation.
The next presentation.
Yes, next presentation.
Yes. Okay. All right. The next person is should be Hun right now. He needs to come back online.
So now you have two more presentations.
Yes.
Okay.
Yes, two more, but then the slides are not with me. My students will be doing the presentation.
Okay.
I need to get across to them.
Mm-hmm.
So I suggest that we take Bukola Kayode so that the next person can join later. So Buki, over to you. You start your presentation, please.
Okay. I'd like to share my slide now, please.
Dear Bukola, could you please, if possible, during your presentation, could you please keep your camera on, if possible?
Sorry, I'm having a bit. No, it's not this one.
Dear Bukola, your microphone is turned off. Yes, now.
Are you able to share your screen, Buki?
No, not yet. I need to send it again. Sorry. Can
Okay,
you go to the next
what,
person,
can I share it for you?
Yes.
I've made some changes, so
can you send
I'll
that
need
to
to
me
share
then?
from
Can you send it to me?
Okay, okay.
You can forward it to my email, then I'll just share.
Okay.
We apologize for this delay. We were sorry for this delay.
No
We will
problem.
get it sorted
No
soon.
problem.
Thank you.
Everything is possible. Technical problems, always we meet with some problems. By the way, if dear Nwadi, if you are ready, we can listen to you. Dear
Yeah, true.
Oluchukwu Nwadi, are you ready? If you want, we can listen to you. You can present. Okay? Dear Dr. Samson, is it possible?
Yes, I am.
Yes, yes, we can allow her to present while we sort this out.
Thank you very
She
much.
can.
Thank you very
Yes,
much.
she can go ahead.
Yes. Dear Nwadi, please introduce yourself briefly, then you can.
Okay. Okay, thank you. Okay, can you see my screen now? Hello?
Yes, we can.
Can you hear me?
Yes,
Yes, we can.
Hear and we
We
see.
can.
No problem.
Okay.
I hear you.
You can see my screen also. Okay. I'm presenting a paper titled Snacking on Whole Wheat Flour and Chicken Egg-Based Extrudates and Consumer Behavior. I'm Wadi Oluchukwu Margaret Mary, the corresponding author. The paper is by Wadi OMM and Okonkwo TM from the Department of Food Science and Technology, University of Nigeria, Nsukka. Introduction. Snacks have become an important part of the human diet, mostly because it is highly consumed all over the globe. Bawa and Seed stated that it is difficult to give snack a definition or categorize snack. However, describe snacking as consumption of products which are easily handled, small, solid or liquid, hot or cold, with little or no preparation methods, and usually used to satisfy momentary hunger. Snacking is very common in developed countries and mostly among young adults. Snack can also be made to be rich in nutrients, depending on the preferences of the producer and or consumer, as the case may be. As a result of the high consumption of snacks, the nutritive value needs to meet certain requirements, especially because some people have replaced snacking with conventional food so as to help them minimize consumption of junk. This paper aims to evaluate the sensory quality of the extrudates from whole wheat flour and chicken egg. Materials: 4 bags of 50 kg each of whole wheat flour, we purchased from Northwest University Farm, Mafeking, South Africa. Preparation of extruded egg snack. The egg whites were separated from the egg yolk for samples requiring either egg yolk or egg white using an egg separator. The whole wheat flour and raw whole egg or raw egg yolk or raw egg white were mixed using a 50-liter paddle mixer constructed by CFAM in Potchefstroom, South Africa. This is the paddle mixer. That's the paddle mixer in use. This is the whole wheat flour in the mixer, and this is egg white. This is the mixer also in operation after putting the egg white here. Sorry, I lost connection.
No problem, no problem. There's a connection.
So I was on this table. So these are my samples.
No,
And I used
table
a
one.
twin screw extruder. Can you hear me? Yes, table one, my formulations for the 7 samples as shown. I use this extruder, the TX32 extruder, twin screw extruder, produced by CFAM, Center for Advanced Manufacturing in Potchefstroom, South Africa. That's the extruder. It's feeding the mixture into the hopper here, as shown. That's the extruder, the hopper of the extruder in operation. Then the extruder, and that's the sample coming out of the die, as shown there on the extruder. That is it. In CFAM, Potchefstroom, South Africa. Then, the stray parameters were constant for all the samples. As I produce my different samples, I maintain the same parameters. Look at the production processes as shown on this flow chart, as I described earlier. Sensory evaluation of the samples was done. The samples were evaluated for sensory acceptance using a 15-man semi-trained panel members made up of staff and students of the Department of Food Science and Technology. The evaluation was by assessing the degree of preference of the different samples in terms of color, texture, appearance, flavor, taste, and overall acceptability using the 9-point hedonic scale shown below, where 9 presented likely, like extremely, and 1 presented dislike extremely. So my data was analyzed using a one-way ANOVA in a completely randomized design. This operation was by Duncan's new multiple range test. Simulation was assessed at P less than 0.5. So this is the sensory evaluation room. A sensory evaluation in progress with some students, semi-trained panelists. That's it, still ongoing, the sensory evaluation. Results and discussion. The color of the control sample, that's that having 100% whole wheat flour, was brown. Wheat flour is naturally brown. The way heated as in baking or hot extrusion as I did, further browning takes place, especially on the surface, resulting from the caramelization and Maillard reactions, as you can see. This is the sample with 100% whole wheat flour. When substituting with whole wheat flour, with raw whole egg at 15%, it became darker. This could result from the protein and lipid reactions, that's the Maillard reaction, to cause the browning. There's already protein in the whole wheat flour, so adding the egg increases, so made the product even darker. So adding egg to whole wheat flour produces darker products. That's dark brown. The intensity varies depending on the quantity of the egg added, and if it is the whole egg, the raw the raw whole egg, the raw egg yolk, or the raw egg white. Samples having the whole egg and the egg yolk seem to be similar, which means that the egg yolk makes the samples darker than just egg white. The samples having egg white were lighter compared with those samples having either whole egg or egg yolk, as can be seen. And the samples having this egg white had stringy appearance. This one is 15% egg white, while this is 20% egg white. So we can see that the stringy appearance for having 20% egg white. This could be as a result of the high protein content in the egg white and dilution. Egg white contains more moisture than whole egg or egg yolk. So that must have diluted it and led to the lighter color.
Hello, sir.
Hello, I'm here. I think there's
I
a
think
connection.
we lost the connection again.
Yes,
Okay.
yes.
Can we then take somebody else, or maybe
Yes,
because
sure.
our time is running very fast.
Yes. Yes,
Okay, maybe we should.
Samson, if you want, your student can present
Okay,
your study.
Toyosi, are you ready? Toyosi can start if he's. I think he's online.
Okay, I'm ready.
Please share your screen. Thank you.
Okay. Is my screen visible to everybody?
Yes.
All right. Good afternoon, everybody. Good morning. My name is Toyosi Timileyin George. I am here to. Okay, I'm currently a master's student at the Cape Peninsula University of Technology, Western Cape. South Africa. And I'll be presenting briefly on a review paper, Potentials of African Yam Bean for Food and Industrial Use. A review. The work was done in conjunction with Dr. Samson Oyinka and Dr. Antonio Bilano of the University of Johannesburg and Cape Peninsula University of Technology. So I move to the outline of the presentation. So we have the introduction, the objective and justification of the review, overview of literatures, just a few, presentation of data from previous studies, some findings that were made, conclusion and future perspective, references and acknowledgement. So I start with the introduction. Just to describe what African yam bean is, according to how it was set up here, African yam bean is an underutilized, hard-to-cook pulse crop, which is native to West Africa and some part of East Africa. Over the years, researchers have found out that and have proposed that it is an important food security crop. And then it has also been used over the years in the fortification and enrichment of other foods, such as commonly consumed staple foods all around. Also, researchers have mentioned that it's a source of healthy starch for use in the food industry. And then it has also been proposed for use for consumption at the household level. So objectives and justification of this study. Okay, we found out that there were few or no up-to-date reviews on the beans as at 2020 when the work was carried out. Also, the information on the proximate composition of the bean and the product fortified with it, we saw that, okay, there were information on it. We had to put it together and share it as a literature. Also, we found out that the potential of the bean in the production of starch and its complexes for industrial use has information that need to be disseminated out. And then we also found out that even though it's an underutilized crop, it is an important food security crop, which makes us also to embark on this study. We also discussed and we saw that, okay, we can elevate the status beyond the household use because it is commonly used in the rural areas. Overview of some of the literatures, just a brief. So we saw that the proximate composition of the bean, notably the protein composition here, has about 19 to 30% of protein, and it compares favorably with some commonly consumed pulses such as pigeon pea, bambara groundnut, chickpea. Although it's not on the screen here, but we also found out that the carbohydrate content is high, and it ranges from around 52% to 68.5%. Also, we note that its underutilization, according to literature, can be ascribed to its hard-to-cook nature and the presence of non-nutritional compounds. Non-nutritional compounds, they disrupt normal metabolism of the bean when consumed. So we found out that this is one of the reasons why they are still underutilized. Other reasons why they are still underutilized is when they are consumed, they cause bloating and flatulence. So we also note that from literatures, the bean has been used in the fortification and enrichment of different products, such as breakfast diets, winning and instant diet, snack and cookies, and then traditional foods. So here I have some information on some staple foods that have been fortified and enriched with African yam bean. For example, African yam bean was fortified with cookies, and up to 40% AYB was used. We have about 38% increase in protein, which is a significantly higher amount needed. I mean, among the consuming populace of cookies, generally children of age 2 and above, they consume cookies, and even adults consume it. So it's actually a source of protein. The normal cookies we consume as humans, it is not as rich in protein as so we also saw that the fortification of AYB up to 40% actually cause an increase in protein. Also, another author worked on the fortification of AYB with up to 40%, fortification of cookies with up to 40% AYB and with cocoyam flour, and there was about a 53% increase in the protein contents. Also, we looked at it's used in the fortification of maize blend, kokoro. So kokoro is a traditional snack that is consumed among the Yoruba, the West African people in southwestern populace in Nigeria. And this actually worked with AYB in different proportion. And we saw that at optimum condition, about 40% also, there was a 32% increase in protein. So we saw that AYB actually, even though still remains underutilized, it can become an important fortification ingredient in the food industry. Other authors that have worked with fortification and enrichment of AYB are Babarinde et al. That worked with fortification of AYB with OFS, fortification of OFSP with AYB for breakfast diet. And then the result yielded about over 100% increase in protein when it was fortified with close to about 30% AYB. So it shows that when you consume this kind of breakfast diet, you are getting enough protein that can sustain you for the day. Also, we note that in the preparation of a complementary diet between AYB and sorghum, we noticed about 76% increase in protein. The work was done by Egbuji and Okoye in 2015. Amala was enriched with AYB2 with African yam bean, and there was approximately about 30% increase in protein after it was done. Also, up to 40% African yam bean was used in the enrichment of tuwo. Tuwo is a staple that is consumed mostly among the Hausa populace in Nigeria. And the author stated that there was about 130% increase in the protein content after about 40% enriched. After about 40% AYB was used in the enrichment of tuwo. Also, there was in the production of agidi, the production of ogi, and various other foods. The bean has been used as a fortification and as an enriching ingredient. So we looked at processing of AYB. So we saw that, okay, processing is done actually to increase the bioavailability of the nutrients in the plant, in the crop. Also to improve the quality and the functionality of the crop. For example, now, AYB, because of its hard to cook nature and because of its problem of anti-nutritional factors, has been proposed in the production of starches. So processing of AYB into starches actually improves the quality and the functionality of the bean as a whole. Also, the reduction of anti-nutritional compound. Processing is done to actually reduce to the barest minimum of the anti-nutritional compounds. Also, it's not stated on the slide here, but I want to say that some processing operation that has been noted in the reduction of anti-nutritional compounds of AYB includes germination, roasting, fermentation, hydrothermal treatment, and soaking prior to cooking. Also, we also noted that processing is done for ease to lessen the effects of the hard to cook property. For example, in a study carried out by Ojo and Adiomowaye in 2015, the author noted that soaking the bean for about 12 hours before cooking actually lessens the cooking time and reduces consumption of energy that is meant to use to soften it. So then we actually move to the important findings from literature that can help people from the review. We saw that AYB can be used in the production of starches for industrial use. For example, an author noted that, Mafosa and Gideani noted that starches from pulses can actually become an important industrial starch because they are healthy and they can be used among people with lifestyle diseases in the production of their diet. Also, we also noted that the starch yield from AYB is about 40%, according to a study carried out by Adebowale in 2009. Also, we also noted that the content of lysine and methionine in AYB seems higher than in most other pulses. This was stated by Evans and Eisma, 1979, and Adebowale et al. 2009. Also, lysine and methionine actually are they are essential amino acids that are not present in some in some cereals, and so AYB can actually improve the inclusion of AYB can actually help increase the presence of these essential amino acids. We also noted that the phytochemical data of the bean was actually was actually researched on by Obo et al. 2009, Shueton 2018, and Emujegba et al. In 2015. And the data indicates that AYB is a potential candidate in the development of functional foods, as they were able to get substantial amount of total polyphenols, total flavonoids when different solvents were used in the extraction of this compound, of these important phytochemicals. Also, we noted that development of AYB fortified and enriched diet still pose some limitations with sensory preference, as sensory data from findings indicates that some of the consumers are not really satisfied with the sensory profile. And so we propose that the development of an optimal design of a design experiment that can give us an optimal condition, optimal amount of AYB that can be used as fortification ingredient in such a way that the amount of nutrients that it adds to the initial staple does not diminish, and at the same time the sensory profile and the preference still remains acceptable among consumers. If we have this data available, this can be used in the industry among don't know why it cut me out. Okay, so I'm sorry for that break. The development of, also we noted that the limitation to the use of the bean is largely ascribed to its hard to cook attributes and the presence of anti-nutritional compounds that has been mentioned earlier. And then, so I moved to, those are the important findings from literature. They were summarized because of the time and every other constraints that we have. So we concluded that the review carefully presented AYB as an important food fortifier and enricher, even though it is still largely underutilized. Also, we said that with the possible negative effect of climate change, the bean can become an important food security crop because it can grow in areas that there is not enough water for growth. They are drought tolerant, just like other pulses like Bambara groundnut. Then we say that its use and status still remains at the household level. So we are looking at a way of elevating their status beyond their use among the peasant farmers that produce it. So also we are proposing that more awareness about the crop be embarked upon by extension workers and government bodies saddled with food security and sustainable development. So we are moving towards an era in such a way that there might be challenges with food security. And then we are proposing that government workers, researchers should also help give information and also recommend this bean as an important food security crops. And this can not only be done by food scientists alone, this can also be done with extension workers, agricultural scientists, and other professionals. So we also recommend that the development of AYB meat analog and non-dairy milk should be intensified to mitigate over reliance on animals and also popular legumes that are expensive. For example, now the major legume that is used in the production of milk is soybean. So we are proposing, researchers have actually found out that, okay, AYB can also be used. So we are saying the industry can also take its use into consideration in the production of non-dietary milk and also in the production of AYB meat analog for vegetarians that do not consume meat. Also, we are saying that sensory acceptability of enriched, complemented, and fortified diet from AYB can be enhanced using experimental designs. So we are saying that, okay, experimental design software can be used in estimating the amount of this bean that can be used in enrichment, fortification, and complementation, so that it meets the protein content that is desired, and at the same time, it does not diminish the sensory preference. Also, we noted that the full profile of the bean has not really been worked upon. So we propose that the full profile can be determined using LCMS and NMR, nuclear magnetic resonance. Also, we say that in vitro and in vivo digestibility studies can be embarked upon. So by using animal models, such as mouse models. Also, in vitro studies can make use of in vitro simulated gastric models and intestinal models, just to know the digestibility profile and the release profile of the bean when consumed. So these are some of the references that were used while this work was prepared. Just some of the references. There were more, but these are some of the references. And then I want to acknowledge the people that actually supported the work: National Research Foundation of South African Government, Cape Peninsula University of Technology, and I also want to thank the collaborators and the co-authors. Thank you very much for listening.
Thank you very much for your presentation. By the way, dear Bello, Florence Bello, are you with us?
Yes, I think she's in now.
She
in? You know, she's not here.
I think I can't see her.
Okay, maybe we can take Buki and then we come back to Bello.
We can get it.
So Bukola, over to you. The floor is over to you now. George, can you end your screen? Okay.
Okay. You can share for me, Dr. Samson, sir.
So Buki, it's over to you. I think you need to share your screen now.
I can't even find my slide. Can you share for me, sir?
Okay, I'll do that just now.
Thank you.
Okay.
Slide. Hey.
Okay, so we can have Buki. Buki, you can start now.
Good morning, everyone. My name is Bukola Kayode, and I'll be presenting on the topic titled Chemical, Physical, Chemical, and Functional Properties of Flour from Frozen Cassava Roots on behalf of the 3 co-authors, including my supervisor and the second author. Following the outlines on your screen, cassava roots is a very rich crop is very rich in various nutrients and has a high demand due to its domestic and industrial usage as raw materials in both food manufacturing industries and home consumptions in various forms. However, the poor shelf life of cassava roots, the low protein content, and natural occurrence of cyanogens have been the major limitations of the utilization of the root crop over the years. And due to this, several authors have researched on how to extend the shelf life of this root crop by storing in boxes lined with moist sawdust, as well as using jute bags, which are basically traditional methods being adopted previously, to the more recent method of refrigeration and freezing using freezers. Several authors have adopted various techniques in storage of these roots over the years. And Akingbala in 2005 found that trench storage of cassava roots in polyethylene bags produced gari with acceptable physical and sensory qualities, as well as under frozen and refrigerated storage. The pasting properties of starch from these stored cassava roots was found to be influenced by refrigerated storage with no changes on pasting temperature, but increases setback, breakdown, and final viscosity. In 1994, dated back and found out that the moisture content, pH, acidity of products from the same cassava roots, which is gari and fufu, remained fairly constant irrespective of the length of storage of the cassava roots. However, some contradictory reports on the composition showed that the moisture, carbohydrate, and cyanogenic potentials of cassava reduced while the ash, crude fiber increased with storage period. Previous research also noted that low storage temperature for low temperature storage for cassava roots have shown to extend the shelf life of the roots. Why is this work then important? Freezing has been shown to increase the shelf life of cassava roots, but then it can affect the quality of resulting products. Therefore, the need to explore and see the chemical and some other properties of the products called cassava flour from the frozen cassava roots. In achieving this, the main objective of this research is to investigate the effect of freezing on physical, chemical, and functional properties of flour from cassava roots frozen for a period of 28. In doing this, it's important that the color, the pasting, the proximate, functional cyanide, mineral materials for the project was gotten from an institute not far from the laboratory where it was used. Looking at the flow chart, the fresh cassava root was harvested and sorted, washed and drained, chopped into further sliced, and then packaged in low density polyethylene Ziploc, -20 plus or minus 1 degree Celsius, using a blast freezer that was noted to be 30% faster than the usual to obtain the frozen cassava slices. The frozen cassava slices was further dried in the oven at 55 degrees Celsius for 24 hours, milled, then sieved, and then packaged to obtain the cassava flour, which was further analyzed. The analysis carried out with the proximate composition, the color, cyanide, pH, the mineral functional, and pasting properties. Then analyzed using the SPSS for significance at 5% level. From the result, the obvious recognition here is that the frozen cassava flour, the flour from frozen cassava roots showed more of a slight browning compared to the one that was gotten from the unfrozen roots, which could be a physical recognition to them. So further to the proximate composition of the flour, the moisture, ash, fat, and fiber of the cassava flour obtained decreased with increasing freezing period, which could be possibly expected due to the ice crystals that have probably broken down due to the effect of freezing on it, and then further aiding the changes in the composition. However, the slight changes that occurred on the protein content was significant. However, it also decreased from the unfrozen to the 28 days of freezing. Furthermore, was the color or the lightness, and the yellowness decreased as well. But however, there was an increase in the redness value of the cassava flour. The striking point here is the cyanide content, which decreased drastically from between the flour obtained from the unfrozen roots and the flour obtained from the cassava, the cassava flour freezed for 28 days, which was basically a lot of decrease occurring in the cyanide content. The pH content, however, is closely related, which is expected since cyanide is a weak acid. The mineral content, major minerals found in this cassava flour were calcium, sodium, magnesium, and potassium. Calcium, sodium, magnesium, potassium are important minerals in human food. They were, but however, others were found in relatively small quantities. With the exception of potassium and manganese, the calcium, sodium, magnesium, copper, zinc, and iron contents of the flour from frozen cassava roots were lower compared to flour from freshly harvested roots. The flour, as shown here, can be tagged as a good source of potassium, magnesium, sodium, and calcium, whose salts are known to regulate the acid-base balance of the body. Also is the next slide. Next slide, sir. The swelling capacity. Swelling capacity, sir.
I'm there.
The swelling capacity as seen from the graph showed similar pattern, showed similarly different from the control, which is the flour from the unfrozen root. There's a striking increase from 50 degrees Celsius to 90 degrees Celsius. And however, there was a pronounced swelling at 60 degrees Celsius straight to 70 degrees Celsius, which leveled out at 70 degrees Celsius to 90 degrees Celsius. This increase can be associated with starch gelatinization and some of the factors that could possibly affect swelling is not limited to what was done here, but the flour composition, the granule size, variation in amylose to amylopectin of the starch fraction in the flour could also be a contributing factor to the swelling power of the flour. Also, the loose bulk density and the packed bulk density was analyzed. As you can see, there's a striking increase. The packed bulk density is definitely higher than the loose bulk density, and this is most likely expected. However, and this is because the tapping process removes scarce species. Bulk density has been associated with the particle size and measures the compactness of particles. The decrease in the bulk density crystals on the starch content of the roots during freezing, which eventually resulted into reduction in the particle sizes as well. Reduction in the bulk density with freezing could possibly suggest that more economical packaging material will be suitable for packaging of the flower samples that were obtained through in this project. The water absorption capacity and the oil absorption capacity as well. The ability of the flower to imbibe water is showed a higher increase more than the ability of the sample, the flowers, to absorb oil. Freezing did not change the ability of the flower to absorb oil, but it did change slightly the ability of the flower samples to absorb more water into it. The significant increase here can be attributed, the significant increase in the water absorption capacity can probably be attributed to changes in the size as reflected by the bulk density as well. The values obtained in the bulk density, both packed and loose bulk density. Small sizes of particle, however, are expected to absorb higher amounts of water. This could be due to the increased surface area of the flower sample. The pasting property, freezing influenced the pasting properties as well. The peak time averaged up to over 4.23 minutes. Pasting temperature average was as well over 74 degrees Celsius. But generally, flower from 28 days had highest peak, trough, breakdown, final viscosity, and setback viscosity. Inconsistent trend, however, between storage days, which shows freezing may have impacted the internal structure, was observed in the results obtained. The example which can be noted is that peak viscosity is, can you go back, sir? Peak viscosity is, however, influenced by the ratio of amylose to amylopectin, the degree of starch damage, presence of non-starch components. Therefore, it is possible that the ice crystal disrupted the cassava tissue, resulting in significant changes. In conclusion, carbohydrate was a major component of the flower samples obtained. Freezing did not relatively affect the proximate composition, but altered the color. There was increase in the ability of the flower samples to imbibe water. Potassium, sodium, calcium were the major minerals in the cassava floors. Next. Freezing did not affect the pH, but it did reduce the cyanide content. The pasting properties of the flowers were affected by freezing, though no particular trend was observed. Cassava roots may be frozen for 28 days and still be used as flower production for various industrial applications, as well as home usage. Future studies should explore the flowers in baked and other value-added products. These are some references, and I would like to appreciate the Comrade State University and other sponsors for giving us the opportunity to participate in the conference for free on behalf of my supervisor and the co-author. Thank you.
Thank you very much. Thank you very much for your presentation. Anyone has any question? No. Now, I will read the names again from second presenter, Forestry Research Institute of Nigeria. There is 5 persons of a group, study group. Any member of this group with us? No. Dear Florence Bello, now are you ready?
Yes, sir, I'm ready.
Okay. We can go with you.
Thank you. Doctor.
Yes, I'm doing that right now.
Okay, thank you very much.
You can start, please.
Okay. Good day, everyone. My name is Florence Bello. The paper I'm presenting titled Nutritional, Antioxidant, and Pesticide Properties Potential of Optimized Pear Millet Flour enriched with mung bean and integuanol pomace flour. Introduction. Enrichment is identified as an effective tool to improve the physicochemical properties of cereal flour. The reduction of protein, energy malnutrition, and other related diseases, and prevention of other related diseases could be as a result of incorporation of legumes and agro-industrial byproducts. And this has been part of the food formulations, which has been the recent development as nutrient diversification strategy. Pear millet, Penicetum glaucum, are mostly found cereal in arid and semi-arid part of the world, especially in Nigeria. Because is common in Nigeria, but underutilized. This pear millet, please go back. The pear millet, please go back. These pear millet are are good sources of energy, mineral, vitamins, dietary fiber, and polyphenol. And also, it also contains high quantity of essential amino acids and some potential health benefits, has been reported by David et al. 2011 and Isovo et al. 2013. Please move on. Next slide. 2013. Next slide. Mung bean is a legume cultivated for its edible seeds and sprout. And this seed grows well under most adverse-arid and semi-arid conditions. And most interesting part of this mung bean seed, as reported by Kuma et al. 2018, is that it contains high amount of protein content of 24.2%, and also very low fat, which is very rare in other legume seed. It contains 1.30% fat and also 60.40% carbohydrate. Tiger nut pomace is a byproduct, that is the residue obtained from tiger nut milk production. And in Nigeria, this tiger nut pomace always been discarded after the extraction of the milk. And according to Figueroa, is reported that it contains considerable amount of components, including dietary fiber and antioxidants. This paper aims to evaluate some chemical and pasting properties of optimized flour blends. Next slide. Materials and method. Flour is produced according to Pongodi and Jemima 2009.
think we lost her. We lost the voice. She's no longer there. Dr. Mustafa. Hello, sir.
Hello, I'm here.
Yeah, I think we lost our connection.
Yes.
She's getting back now. Yeah, she's back. She's back.
I'm sorry for
No,
breaking.
connection again. She connected again.
All right, yeah.
I am back. Can I continue?
Yes, please.
Can I continue?
Please, yes.
Hello?
You can, you can.
Okay. Okay. Materials and method. Okay, can I continue?
Yes.
Okay, when millet is produced according to the method described by Pongoji and Jemima, 2009, Mumbi flour also is produced following the method that described by Ikarue and Onwuka 2010. And also tiger nut pomade is simply produced by extracting out the milk from the tiger nut, and the residue is what we call pomace. Experimental design used is the optimal mixture design of response surface methodology. And the quality of the flour produced have been evaluated, in form of functional properties, proximate composition, antioxidant. We check the phenolic content, flavonoid, and DPPH, radical scavenging activities of the flour produced. Next slide. Result. 16 runs were generated from the experimental design of reference surface methodology were displayed in table 1. A, B, C, the A denotes pure millet flour. Next slide, please. Okay, thank you. Represent mung bean flour and C represent tiger nut pomade flour. These three are also called independent variables. And according to statistical analysis used, which I call the optimal mixture of response surface methodology, generated these 16 runs. And out of these 16 runs, the highlighted row in blue, run 1, run 4, run 7, and run 13 were selected based on the ANOVA result displayed in table 2. So let's go to table 2. This display in table 2. Table 2 showed ANOVA for functional properties, as I've said. BD represents bulk density, GT gelation temperature, swelling capacity, foaming capacity, water absorption capacity was significant. They were significant in this analysis. Under the bulk density, the independent variable pure millets and the mung bean flour the mung bean flour contributed positively to the flour blend. That's why under the C, which is the tiger nut pomade, the negative sign there represents that the tiger nut pomade contributed, contributed negatively into the flour blends. And also the gelation temperature here we show 153.11 under tiger nut pomade. It contributed greatly to the gelation temperature of the flour blend. And also they say for water absorption capacity too, both independent variables, the mung bean and the tiger nut pomade use also contributed well to both the foaming capacity and water absorption capacity. Whereby under the whole absorption capacity, the tiger nuts really affected the way the flour blend, the way the flour absorbed the hull. That is why it showed the negative sign. Next slide. Table 3 shows the proximate composition of the optimized bare millet, mung bean, and tiger nut pomade flour blends. As I've said, the 4 the 4 runs were selected based on the good functional property that I've said, water absorption capacity and the bulk density and the swelling capacity. So this for run 1, run 4, run 7, and run 13. Out of these runs, it was observed that run 7 had a higher protein content of 15.57%, and also the hash content also was also high, but not significantly different from the run 4. But for run 13 also, it was also observed that the crude fiber was also high and energy value. And this is as a result of the incorporation of the tiger nut pomace flour. Next slide. Table 4 also shows the antioxidant activities. Under this, we determine the total phenol content, total flavonoid content, and DPPH. And from the figure, it is seen that run 13 that contain 85% pear flour and mung bean 5% mung bean flour and 10% tiger nut pomace flour, that is run 13, emerge as a higher TPC, that is total phenol content, compared to other blends, followed by run 7. And also the same thing, total flavonoid content here, the same run 13, also as a higher, emerge the highest compared to other run, and also for DPPH. And it was also observed here that the pear millet, because looking at the composition of the run 13, it was shown that the pear millet 85%, and also from the record that the pear millet has a higher content of antioxidant and properties. Also, we're also talking about the pomace from the tiger nut. They also have the higher content of antioxidant properties in terms of the total phenol content, flavonoid, and also the DPPH. The next table, table 4. The pasting property of the optimized pear millet, mung bean, and tiger nut pomace flour is displayed in table 4. Here, run 1 was highlighted red, which shows that it has a low peak viscosity. In fact, in all the parameters analyzed under pasting property, run 1 that contains 70% pear millet flour and also 20% mung bean and 10% tiger nut pomace flour had the least peak viscosity value, trough viscosity breakdown, final viscosity setback, and also the setback viscosity of 98 CP. And also, when we look at the run 7 has the higher peak viscosity of 280.50. In fact, all the parameters analyzed, also run 7 emerged the highest compared to the other run. Though, when we convert these centipoles to normal RVU, rapid physical analyzer unit, we will discover that this run 7, 280.50, when we convert it to RVU, is also equals to 28.0, which means that it's very, very low. So all the pasting property analyzed here, they are very, very low compared to the literature, the one found in the literature. And also this can also will not be far-fetched, because when we look at the pear millet itself, which is the main ingredient in this flour blend, I've always been using for complementary food, because of the low peak viscosity and also because of the breakdown viscosity. That is why I almost use for the complementary food. And also, apart from complementary food, you can also as well use it for any food that require the low viscosity. I mean, in terms of texture. We're talking about those confectionery food that require low texture, and the pear millet will be also as well good for the preparation and as a major ingredient. In conclusion, the research has shown that the blending of pear millet flour together with flour and tiger nut pomace flour using optimal mixture design of response methodology will enhance the functional and nutritional properties of product requiring a low viscosity. And as you've seen, there are on 7 that contains 73.75% pear millet, 20% of mung bean, and 6.25% tiger nut or maize flour, and as well also the on 13 that contain 85%, higher content of the pear millet, 5% of mung bean, and 10% tiger nut or maize flour, emerge as the one that contain higher protein content, fiber content, and also as well as the antioxidant activities, the one that would determine. So the use of the traditional crops will reduce over-dependence on wheat, and that is why we combine our local crops. Pear millet is locally sourced for, and also the mung bean flour, and also the tiger nut for maize. So this research finding also encourage the use of agro-industrial based products, which are currently not fully exploited. We also want to appreciate the organizer of this conference for the opportunity given to us to present free. Thank you very much for your attention.
Thank you very much, dear Florence Bello, for your presentation. Now, dear Abo Fadeju Jonathan, welcome. How are you?
Dr. Mustafa, I think Nwadi was presenting before. She stopped along the way. Maybe we're allowed to complete us.
Yes. So,
Nwadi can continue.
Dear,
Okay, thank you.
now our last presentation, Baba Yejo from University of Ilorin, they are not.
She's not available. She's not available. Yeah.
Yes, and I think it's okay now. By the way, I'd like to share my screen with you.
We have 2 more persons. Doc, we have 2 more persons to present.
Excuse me, you have 1 more presentation?
2 more persons are online to present.
Excuse me. Which presentation you have, dear Samson?
Not me. Nwadi Chuku.
Now, according
That's
to my...
me. I lost connection when I was presenting.
Okay.
And then the last person, Jonathan, is also around now.
Okay.
So they have to snap it because of time.
Okay.
So Nwadi, over to you, please.
Yes.
Okay.
We're waiting for you, madam. Have you found this slide?
I'll be with you shortly.
Dear Nwadi?
Yes, I'm here.
Yes. Okay. Now, actually, there's a problem that now after us, the new group will join this meeting room. Unfortunately, we need to stop. We need to finish to our presentation, our session. Now, just now I'm writing. Please check the chat. Now I'm writing some links there. Please note them. Now, we have another conference, Castro Diet conference. Please check it, and you can join that conference free. For this conference is free for African participants. And after 2 weeks, we have another conference, Agence Journal conference. It's also free for African conference. Its link also I wrote it, agencecongress.org. And I'd like to thank you to all participants. Now we need to leave the room. Thank you very much for your participations. Just there is one author with us, Abo Fadeju. Are you hearing me? Yes. Now we will transfer you to another meeting room to present your presentation. Okay. Now, please leave the room and we will talk with you about your new session because you late and we checked your name several times, but you late. Then now we need to finish the present session and we will transfer you another room that you can make your presentation. Dear participants, thank you very much for your presentations, for your contributions. And until tomorrow, you will receive your certificates. And please check the conference calendar. The conference proceeding books will be published according to the calendar. Thank you very much. Have a nice day.
Thank you.
Thank you.
Candos, are you here? Are you with us? Candos or Atabek, are you with us at control?
Yes, Dr. Yes, Dr. Mustafa.
Hello, how are you?
I'm fine, thanks, and you?
That's great. Now I'm waiting for a participant. Let me check. Where is she? Then I will inform you. Okay.
Okay, of course. I'm waiting.
Dear Jonathan, please turn on your microphone. Can you hear me, Abu Fadejo? If you can hear me. Dear Abu Fadejo, are you hearing me?
I can hear you, yes.
Yes, okay. Now, in case we accept your papers as presented, you need to read the abstracts to me here now. Is it possible? Because we don't have time now, and there is no another space room. If you can read your abstracts, you have, I think, two presentations, two papers. If you can read two abstracts to me, we will accept it as presented, and we will send you your certificates, okay?
Okay. Should I go on? Hello, can you hear
Hello,
me?
yes, yes. I'm listening to you.
Can you hear me? Can I? Okay.
Yes,
Thank
I
you.
can hear you.
My name is, okay. Yeah, my name is Awo Fadeju, YMC Foluke. And I would like to speak on phytochemical screening, antioxidant properties, and microbial indices of extracts from propolis collected in Forestry Research Institute of Nigeria. As he said, I should read on the abstract. Now I'm reading on the abstract.
Yes.
Propolis consists of a complex mixture of resinous substances collected by honeybees from different plant sources. However, the objective of this study evaluated the phytochemical, biochemical characterization of isolate, microbial analysis, and antioxidant properties of propolis collected in Forestry Research Institute of Nigeria as potential bioproducts for food and pharmaceutical industries. Propolis was collected in apiary, the Forestry Research Institute of Nigeria, and it was extracted using hot water and ethanol with variation in time of 36 and 72 hours. Propolis ethanol extract showed highest amount of phytochemical constituent in 72 hours, while the extraction time compared to hot water extraction. It was observed that alkaloids, saponin, phenols, and cardiac glycosides were present abundantly both in the extraction medium. Ethanol extraction of 36 hours showed with the probable isolate of Lactobacillus salivarius and 72 hours yield 17 biochemical acids with Lactobacillus fermentum, while hot water extraction of 36 hours exhibited 16 acids with Streptococcus of aeruginosa in 70% butylated hydroxytoluene 2%. Extraction. Tocopherol, which is a vitamin E, was higher in propolis ethanol extraction with values 19.96 and 26.04 microgram per mil in comparison with hot water extraction. The microbial indices are within the standard. The results pointed. Then conclusively, you can say that the results of the data and some, thank you. Can you hear me? I'm Dr. Mustafa. Dr. Mustafa, can you hear me? Can you hear me?
Dear Jonathan, I think there's a connection problem. Sometimes your...
Seriously, the connection is bad.
Yes. Do you have any connection problem?
Yes.
Okay.
The connection where I'm living is bad.
Okay. Let's do it. Okay. Now, as the moderator, I accept your two presentations as presented. Okay, two papers. Thank you very much for your participation. Now I'm writing to conference coordinator that they can prepare your certificates, all your authors. I think at your first paper, you are 5 authors, right?
Yes,
and
sir.
for
Yes,
second,
sir.
you are 2 authors. Okay, I accept it. Thank you very much. Sometimes here also we have some connection problems, but today we have many problems, connection problems. We couldn't connect. Okay, thank you very much. And by the way,
Can you hear me, Dr. Mustafa?
by the way, I invite you to our next conference. It's also free. Okay. And you can join our conference. Now, can you see the links on chat? Please check the chat. Okay. I wrote 2 links, conference links.
Okay.
And now,
And I've copied it.
now
Okay.
can you,
Okay.
now I will share screen. And can you see gastronomy screen?
I can't see anything here.
Okay, and here there is another screen. Anyway, there are 2 different conferences too. One is gastronomy conference and another is also applied sciences. One is in Jordan at the end of this month. You can send your another abstracts and you can join there freely. For our gastronomy conference also, you and your co-authors can join up to 5 papers. It's completely free. Okay, thank you very much, dear Jonathan, for your effort. Nice to meet you.
Thank you,
Okay,
Dr. Mustafa.
bye-bye.
You're welcome.
Have a nice day.
Thank you. Bye. You too.